made up. It is made up of repeating units of the same unit of monomer. What is the monomer of protein?
Acid amino.
Not
Amino acid.
acid, amino acid, right? Okay, so thank you. So amino acid, you guys still remember the structure is NCC, right? Amino acid, they have the why they are named as amino acid, because amino, they have the amino group NH2, okay, and then acid because they have the carboxyl group COOH, okay. So protein is a polymer, is made up of the repeating units of the amino acid. So whenever we eat protein, okay, whenever we eat some food like chicken, fish, egg, when we eat the protein, so the protein will be digested lah in our digestive tract, isn't it? So it will go through our digestive tract. Okay, the digestive tract also known as the gastrointestinal tract lah. Okay, from our mouth and then the esophagus and then to the stomach and then to the intestine. So along this way, all these proteins will be broken down. Okay, monitor. Okay, so here we can see in the slide during digestion, okay, proteins are broken down into amino acids, okay, and then this amino acid will be used for synthesis of proteins and also for so many functions. For example, this amino acid will then be used to produce another protein, okay, and then for body growth and repair, and also oxidize to produce ATP. Okay, you guys remember about cellular respiration, right? Where the cell, what is cellular respiration? What is the purpose of cellular respiration?
To produce ATP.
Okay, to produce ATP, right? So we learned that cell, they use glucose, isn't it? They use glucose to produce ATP, isn't it? But actually, amino acids also can be used to generate ATP, but the amino acids needs to converted into glucose first. Okay, that we have learned gluconeogenesis, isn't it? Okay, so the protein, the amino acids, they must convert to glucose first through the gluconeogenesis. Then they can be used to produce the ATP as usual, or maybe they can just entering the cellular respiration by being converted to intermediates of the cellular respiration. Okay, so that's why it is said here amino acids can be used to oxidize to produce ATP. Okay, so that's why we can. That's why when we eat, all the foods also can be used to generate ATP, including amino acid. Okay, besides that, amino acid also, they can also just now like what I said. Here, they can be converted into glucose through the process of gluconeogenesis. Okay, to generate ATP, and also they can also can be converted into fat, triglyceride. Triglyceride is fat. Okay, it's a kind of lipid. Okay, so the liver then, now we want to focus on the liver. Okay, because we are learning the structure of liver. So we want to focus on the function of liver for the metabolism of the amino acid. So we have two, which is anabolic and also catabolic. So anabolic is anabolic is the process where we build up from smaller molecules to big molecules. For example, we want to produce protein, so the formation of amino acids to form the protein that is anabolic. So when we say catabolic or catabolism, that is actually we breaking down the large molecule to simple one. For example, we have the protein, so we break down the protein into the amino acids, and then the amino acid is used to convert it into glucose, and then cell use the glucose to generate ATP. That is catabolism or catabolic. Okay. So all the proteins or amino acids that have already been digested through the digestive tract, they will enter the liver. Okay, so the liver, they will further metabolize all these amino acids. Okay, so that's why we are going to learn about the transamination and deamination processes. These are the two processes that will be conducted by the liver to break down. They may be breakdown or they may be uh, synthesize protein also in the liver. Okay, so liver they will do both anabolic and also catabolic process. Okay, so now we straight away focus to transamination. Okay, amination tu, amination, amine. So you guys remember amino group, so amino group is NH2 here. Amino group is NH2. Trans, you guys remember transfer. So transfer. Okay, transfer. So we transfer an amino group from an amino acid. So when we want to transfer, we must transfer to someone, isn't it? So the one who is transferred is the donor, and the one who is accepting the amino group is the acceptor. So who is the acceptor here? The acceptor here is the keto acid. Okay, it's the keto acid. So the donor is the amino group, okay, the amino group, and the acceptor is the keto acid. Okay, so when the original amino acid gives up its amino group, so they become a new amino acid. Okay. So what is the importance of the transamination? Transamination will allow liver to manufacture non-essential amino acid. Okay, this is important in your exam. Okay. We have two types of amino acid. One is essential, another one is non-essential. Okay, for the essential amino acid, we cannot, our body cannot produce the amino acid. Okay, so we have to, from the diet, okay, like from the diet, we cannot synthesize the amino acid in the body, so we have to take it in our diet. It means we have to eat. We have to eat the beef, fish, eggs, poultry, and milk. Okay, then only we can get the essential amino acids. Okay, so but for the non-essential amino acid, okay, but for the non-essential amino acid, it can be produced in our body through the process of transamination. So in exam, if the question asks you what is the importance of transamination or significance, so you can answer to produce non-essential amino acid. Why we need both non-essential and essential amino acid? Because both essential and non-essential amino acid is needed to make protein. Okay, so for us to make the protein, we need both essential and also non-essential amino acids. Okay, so that's why it's important for the transamination to occur in order to manufacture or to produce non-essential amino acids. Okay, so we go straight away to the transamination process. So you see here this amino acid 1. So this is the donor or the acceptor? Hello? So,
Donor.
okay, good. The amino acid 1 is the donor. It will donate the what?
Amino group.
H2.
Yes, NH2 or the amino group. They will, okay, off your mic. Okay, okay, so the amino acid 1, they will donate the amino group here. So you can see the amino acid 1, one is donate its amino group to keto acid 2. Okay, why it is called keto acid? I want to explain. Keto here means they have the carbonyl CO group, C double bond O group. This is uh, ketone group or carbonyl group. Ketone group or carbonyl group, C double bond O. So that's why it's keto. Acid. Why it is called acid? Because they have the COOH, their carboxylic group. Okay. So when you see the structure, C double bond O, ketone group, and also COOH carboxylic group. So this is an keto acid. So the keto acid. So you see the amino acid here, CCNH2HR1. So the carbonyl group, the amino acid 1 here, they donate the amino group. Now they don't have the amino group anymore. So they become the keto acid. So now they become the keto acid. The original amino acid now they become the keto acid. And then the keto acid here, the one that receives amino group, they will become the new amino acid. They will become the new amino acid. So keto amino acid 1 donates the amino group to the keto acid 2. So the amino acid 1 will turn into keto acid. And then the keto acid will receive the amino group. Once they receive the amino group, then they will become amino acid. They become amino acid. So why they become amino acid? Because now they already received the amino group. So now they have the amino group and also they have the COOH as the carboxylic group. So they become amino acid. So this is the new amino acid that is formed. And then the original amino acid now become the keto acid. Okay. So for the keto acid, actually the keto acid that is common that is common for this process is only three: alpha ketoglutarate acid, oxaloacetate, and also pyruvic acid. Okay, actually we have already learned about all these three acids. Where do we find these three keto acids? Where?
Glycol acid and carboxylic.
Good. Okay, in glycolysis and Krebs cycle. Alpha ketoglutarate acid, also known as alpha ketoglutarate, right? You guys remember this one is the intermediates in the Krebs cycle. Okay. Oxaloacetic acid, also known as oxaloacetate, in ionized form, they also in the Krebs cycle. Pyruvic acid, if you guys remember, this is the last product of glycolysis. Okay, so these are the three acids that will be used in the process of aerobic respiration or cellular respiration to generate ATP. Okay, so these three keto acids are the precursor of the three amino acids that can be considered to be completely non-essential. Okay, so for example, the pyruvic will form the alanine, alpha ketoglutarate will form the glutamate, and pyruvic acid will form the aspartate. So alanine, glutamate also aspartate. This is what we say the newly formed amino acid. Okay. Okay, the majority of amino acid will transfer the amino group to the alpha ketoglutarate to make the glutamate amino acid lah. Okay, so if you can see in this reaction, we have the alpha amino acid here, and then we have the alpha ketoglutarate. Okay, alpha ketoglutarate is the keto acid involved. Okay, so then alpha amino acid will then becomes the alpha keto acid, and then alpha ketoglutarate will then become glutamate. See, so glutamate is the newly formed amino acid. Okay, and then what happened to the alpha keto acid here? The alpha keto acid here is either the alpha keto acid here will then will enter the Krebs cycle and use to generate ATP. Okay, and then the glutamate here, they might be used to synthesize new protein. They might be used to synthesize new protein. This is the glutamate here is the non-essential amino acid. Okay, this is the non-essential amino acid. It will be used to produce another protein or any other function. But for this alpha keto acid, what will happen? They will enter the Krebs cycle. Okay. Okay. So if you can see here, the amino acid here, they have the amino group. Sorry, here, this is the amino group. The amino group, this amino group will transfer to the alpha keto acid. So the alpha keto acid will become the alpha keto acid now receiving the amino group. Now it becomes the glutamate. Then what happened to the amino acid here? The amino acid here will become the alpha keto acid, and this alpha keto acid will be going to the Krebs cycle. Citric acid cycle, also known as the Krebs cycle. Okay, they will go to the Krebs cycle and then they will be used to generate ATP. Okay, another one thing that you have to remember, this is transamination is a reversible reaction. That's why you can see the arrow here is reversible. We have two head here because it is reversible. And then another one is the enzyme. Okay, this enzyme also important. You have to remember in exam. This is transamination process, right? So you guys remember the enzyme is transaminase. we put the ES here as the enzyme transaminases. Transaminases. Because they transfer amino group, so it is called amino transferases. Okay, so it is called aminotransferases or also known as transaminases. Okay, either of these two group or enzyme in exam also correct lah. Okay. Okay, that's all for transamination. Do you guys understand?
Yeah,
Yeah, teacher.
understand.
Yes, teacher.
So understand, very understand or understand?
Understand.
Okay. Okay, so now we go to the second process that will be asked in the exam. That is deamination. Okay, deamination, amine stand for the amino group. D stand for the removal. Okay, so deamination means we remove the amino group. Okay, we remove the amino group. We remove the amino group from where? From the excess amino acids. Excess means berlebihan lah. Okay, excess means berlebihan. Berlebihan means this amino acid they are not being uh, just now when they are digested through the gastrointestinal tract, they are being used for the synthesis of protein, for the repair, for the growth. Okay, but there are protein, there are amino acid that is excess, not being used for all these purposes, so they will entering the liver, and then the liver will settle them. Okay, so what happened to the excess amino acid that are not being used to synthesize protein and so on at the at earlier part of digestion? So they will become broken down and they become deaminated. Okay, so liver cell deaminate liver cell or hepatocytes. Okay, liver cell or hepatocytes, they will deaminate or remove the uh, deaminate the amino acids. That means they will remove the amino group from the amino acids. Okay, so this is called deamination. So deamination, the byproduct will be the ammonia because this one you can see this is the ammonia NH3. Okay, but sometimes it is also can be, you can look it, they will show it as NH2. Okay, so when they remove the NH2, the NH2 will then combine with another hydrogen, that it will become ammonia. Okay, for this structure, it's already combined with hydrogen, so it's ammonia NH3. But then some of the picture you will see they will be only drawn as NH2. They will remove the NH2, then NH2 will plus with another H, that will become the ammonia. So this is considered as the byproduct lah, the byproduct for this process of deamination. And then what happened to the, okay, so if you see here amino acid glutamate, okay, once they remove the ammonia, they will become the glutamate here. Once they remove the ammonia, they remove the amino group. Sorry, once they remove the amino group and the amino group remove becomes the ammonia. The amino acid glutamate here becomes new amino acid, which is alpha ketoglutarate. The keto acid alpha ketoglutarate.
Teacher,
Yeah.
teacher,
Yeah.
the amino acid glutamate is donate
Then
remove NH2 or NH3.
okay, actually, they actually remove. Okay, remove. Sorry, not donate. Remove. Yeah, remove NH2.
So the
Remove
picture
NH2.
is two
Mm-hmm. Yeah,
the
the
picture.
picture.
Ah,
Okay. That's like what I said just now. Some picture they can draw NH2, but some picture they draw the NH2 already combined with H. So like this picture, right? They draw it as already combined with H, NH3. But in exam, we don't say they remove ammonia now. We say the amino acid remove amino group. This one. Wait. Okay. This one. Okay. When if you want to answer in exam, you say removal of the amino group, and then the amino group will be converted to ammonia. Actually it's like that, but now in the picture they can also draw the amino group already combined with the hydrogen and become ammonia. Okay, so in this picture we can see they remove the ammonia, but actually in exam, to be specific, first they will remove the amino group first and H2, then only the NH2 will combine with another H and producing the ammonia. Okay, this is just a biochemistry, a very detailed one. It could be ammonia, it could the picture can draw NH3 straight away, or it can draw NH2. Okay, but in exam you guys write deamination is the process of removing the amino group. Amino is NH2. That is the most important lah. Is it okay, Wani?
Okay.
Okay. So proceed. Okay, so when the amino group, when the glutamate, they remove the amino group, so now they become the alpha ketoglutarate. Guys, you guys remember alpha ketoglutarate used in what process? Alpha ketoglutarate used in what? Yeah, Krebs cycle. So straight away, you guys remember they will go to the Krebs cycle. Okay, one of the function. Okay, they will go to the Krebs cycle. This is one of the function. They can also be converted to other molecules, but at least you guys remember one. Alpha ketoglutarate, you guys remember Krebs cycle. So you guys remember the alpha ketoglutarate can be used, one of the function, it can be used in the Krebs cycle to generate an ATP, 1 mark already. Okay? Okay, then what happened to the ammonia here? Okay, the interesting here is the ammonia. Ammonia here is if in high concentration, the ammonia will be toxic. Okay, so if it is toxic, do you want to keep it in our body?
No.
Okay, we don't want to keep it in our body. Only one answer. Babena Sangita, where are you? Are you here?
Yeah, teacher.
Okay. Thank you. So once we have the ammonia here, we don't want to keep the ammonia because the ammonia is toxic. It's a waste. Okay, it's a waste. So liver cell, which is the hepatocyte, they will convert this ammonia into what we say urea lah. Okay, why our body needs to convert into urea? Because ammonia, if it is high concentration, it will be toxic. So if it is toxic, it is bad. It is not good for our body. So the ammonia will convert it into urea. So urea is less toxic, and then urea, if high concentration, also not toxic. They will be harmless. Okay, they will be harmless. That's why we can collect urea in high concentration. Then it's good enough for our body. It's not. That's why the liver will convert the highly toxic ammonia into urea, and then this urea will then will be excreted out in our iron chain, which is the urine. Okay, which is the urine. So that is how we actually remove the nitrogenous waste. Why we say nitrogenous waste? Because we have the nitrogen group here. So ammonia is the nitrogenous waste. So it will be converted into urea. Then this urea will be excreted, dikeluarkan from our body through the urine. Okay, through the urine. Okay, so we will discuss further lah on the urea formation after this. Okay, so just to reinforce that, so in the above example, removal of amino group. Okay, you see in exam, don't say removal of ammonia, say removal of amino group. Then only this amino group will convert it to
Amino
ammonia. Amino group converted to ammonia once it's combined with hydrogen. Okay. Then this ammonia converted to
urea.
Urea, and then be excreted in the urine. Okay. Then removal of glutamate, removal of amino group from the glutamate becomes alpha ketoglutarate. Alpha ketoglutarate will enter Krebs
Krebs cycle.
Krebs cycle to generate ATP. One more. Okay. Okay, so that's all. Then the liver will release the, okay, once we have the urea, okay, the urea, they need to be excreted in the urine, right? So who makes the urine? Who produces urine? Who produces urine? Which organ produces urine, class?
Kidney.
Yes, kidney. That's why this urea need to be transported to the organs that produce the urine, which is the kidney. So this urea will be transported to the kidney through the blood. So the liver will release the urea into the blood, then the blood will go to the kidney, and then the kidney will then filter the blood and then take up the urea. And then at the end of the day, the kidney will produce the urine, and the urine containing the urea, then the urea will be excreted out from our body. Okay, so that's how we actually remove the nitrogenous waste. Initially from the ammonia, and then lastly becomes the urea. Okay. Okay, now we want to focus on what happened to the alpha ketobutyrate here, or the keto acid here. Okay, so just now I said the keto acid here, it can be one of the function, it can enter the Krebs cycle, right? But that's not only actually the function of the keto acid, there are more. Okay, so the keto acid here actually they can be involved in several functions, not only entering the Krebs cycle. Okay, so if you see in this diagram here, uh, so if you see in the in this lecture, it says the keto acid, then the keto acid just now, for example, the alpha ketoglutarate, right? Okay, they may be converted into glucose. Okay, and then they also may be converted into fatty acids, and then they also may be converted into ketone bodies. Okay, and then all these can be used as fuel. When we say as fuel, fuel ni adalah sumber bahan api. Sumber bahan api means this is the uh, for the generation of ATP lah. Okay, it's either to generate ATP through the process of cellular respiration, or they will be just starved. Okay. Okay. So if we okay, let's see the picture here. Okay, let's see the picture here. So the amino acids here. First we have the catabolism and anabolism. Okay, all these amino acids after being digested. For anabolism, they are used to synthesize all the proteins, hemoglobin, and so on. And then catabolize, they are being broken down, and then the excess one will enter the liver. Okay, so if you see here deamination will produce the byproduct ammonia. Ammonia will convert it into urea, and then urea will be released from the liver and then transported to the kidney, and then the kidney will excrete out. Okay, now what happened to the alpha keto acids? If you see here, we have 3 arrows here. Okay, first one, the alpha keto acid, they will be converted into acetyl-CoA, and then it will be converted into ketone bodies. Okay, so here this is the ketone bodies, they can be converted into ketone bodies, and the ketone bodies also can be reconverted back to acetyl-CoA, and then this acetyl-CoA can be used, can be broken down into, can be converted into fatty acids. Okay, and then the fatty acids will combine with the glycerol. Okay, and then fatty acids plus glycerol, then we will become the triacyl glycerols. Okay, so the triacyl glycerol, this is actually fat lah, and then this is fat cell, or also known as the adipose cell. So this fat cell, if you can see here, it is being stored. If adipose tissue is being stored, that's why it stays here. It says in the, it says here, the keto acid converted into the fatty acids, their lipogenesis for storing. Okay, so this is the example. They become the fat, and then they are stored in the adipose tissue. Okay, okay, that's another. So there are 2 functions here that happen to the alpha keto acid. They are converted into ketone bodies. They can be converted into fatty acids through lipogenesis, and then once they convert into fatty acids combined with glycerol, they become the fat, and the fat can be stored in the adipose cell. Okay, besides that, you can see here alpha keto acids, once they become the fat, just now the fat is being stored in the adipose tissue. But they also they can become the fat, and then the fat can be used to enter the cellular respiration. The fat also can be used to enter cellular respiration to generate ATP. Okay, so the fat either you store in the adipose tissue, or the fat you can use for become the fuel for the cellular respiration to generate ATP. Okay, and then besides that, alpha ketose acid also. Just now we have the alpha ketoglutarate, and also we have the some of the amino acid can be converted into pyruvate. So if you guys remember, of course, pyruvate and alpha ketoglutarate, these are the intermediates of the aerobic respiration. So that's why it's written here arrow. It's written the arrow here. It enters the cellular respiration to generate ATP. Okay. Okay. So let's read this. So carbon chain of all the amino acids are metabolized and used as reactant in one of the steps of aerobic respiration to generate ATP. Okay, so during deamination, for example, this one is an example only. Alanine converted to pyruvate, glutamate converted to alpha ketoglutarate, aspartate converted to oxaloacetate. Okay, so then the pyruvate, the alpha ketoglutarate, and the oxaloacetate, they will then enter the cellular respiration. So if you see in the diagram here, we have glycolysis, we have the Krebs cycle, citric acid cycle, also known as Krebs cycle, and then we have the ETC and also the chemoosmosis. This one where we receive the NADH and also the FADH2. So you can see the protein, right? The protein here they are broken down into amino acids. Okay, then this amino acid they are converted into the pyruvate, the acetyl-CoA, and the oxaloacetate, and alpha ketoglutarate enters the Krebs cycle. And then the byproduct is and then the byproduct is ammonia, will be excreted in the urine. Okay. So for your information, all the amino acids also they can be used as the reactants for the aerobic respiration. So this is the overall picture, just like this now. This is the pyruvate acid, right? Pyruvate acid, or known as pyruvate. Okay, and then this is acetyl-CoA, and then this is oxaloacetate. This is alpha ketoglutarate. Okay, and then this is fumarate, and then this is succinyl-CoA. Okay, so this whole thing is Krebs cycle. Okay, and then this is the okay, so this is the glycolysis. Okay, so you see all these amino acids, they will be converted into the intermediates of the aerobic respiration, which comprise of glycolysis and also Krebs cycle. Okay, so they all have to go through the process of deamination. Okay, they have to remove their amino group first, then only they will be converted into pyruvate acid or acetyl-CoA or oxaloacetic acid, fumarate, and so on, and then they can proceed to aerobic respiration to